In this lecture, we are going to discuss another very important technique which is used to determine the heats of reactions in which the amount of heat absorbed or evolved is very small. And that technique is called isothermal titration calorimetry. As we earlier discussed, that calorimeters have been there in the market since 18th century. However, with the time, the technology developed, and it was always felt that we should have calorimeters available which are capable of measuring the heats of reaction of the order of microjoules. Most of the biological interactions taking place in the living systems are weak. The heats of interactions can be very small, and therefore, if we want to address those phenomena, those interactions, we need very sensitive microcalorimeters. Earlier we discussed differential scanning calorimetry, and today we have differential scanning calorimeters which are very sensitive, which require only a few micrograms of a sample to give the values of the transition temperature, enthalpy of unfolding, entropy of unfolding, and other thermodynamic quantities to a very good degree of accuracy and precision. And now let us discuss a technique which can give us how much heat is released or absorbed when two species or more species are interacting. Let us take a look at this slide. And we just start with an example of A plus B forming C. And whenever we talk about this kind of reaction, the questions can be, can we answer qualitatively how this is happening? Qualitatively means, okay, A is mixed with B and it forms C, and qualitatively we establish that there is A, there is B and there is C in the mixture. And when we talk about quantitatively, quantitatively means we must assign the numbers. Here particularly if I talk about this reaction, in order to make quantitative, I must be able to tell how many moles of A are interacting with how many moles of B and how many moles of C are being formed. And secondly, how much heat is liberated or how much heat is absorbed when this reaction takes place. And the third thing is, what is the equilibrium constant of this process? And if I know the equilibrium constant, I can immediately get the change in Gibbs free energy, standard Gibbs free energy. And if I know now the heat, if I know delta G naught, I can calculate delta S naught. That means, by making it quantitative, I am talking about establishing the stoichiometry of the reaction, establishing the value of equilibrium constant of this reaction, determining or measuring the values of heats of interactions and entropy change. Once I have all these thermodynamic quantities, I can describe the process both qualitatively and quantitatively. And then when I talk about the interactions, when two species are interacting, there is some amount of heat is either released or absorbed. And there are certain interactions which release heat, and certain type of interactions absorb heat. And therefore, if we are able to measure the heat changes very precisely, that means we can connect the heats liberated or heat absorbed with the nature of intermolecular interactions. And these things we will be discussing in details, which are of tremendous academic and industrial importance, and we will take up in the future lectures. Isothermal titration calorimetry today has become a popular choice amongst the biologists, biochemists and chemists who are interested in understanding various types of interactions in which the amount of heat change or the heat liberated or absorbed is medium to very low. Why isothermal titration calorimetry is important? Let me just read out some of the areas where the isothermal titration calorimetry has been applied very successfully. Typically, the binding constant, whether you write Kb or you write Ka, Ka is used for association constant, Kb in literature many times Kb is used as binding constant. Physical chemists would like to express in terms of Ka association constant or Kb or binding constant, or you can simply say K. Biochemists prefer to express in terms of dissociation constant Kd, and we know that Ka and Kd are reciprocal of each other. So, therefore, we should not be confused when we are talking about Ka or we are talking about Kd. Whether we talk about K or Ka or Kb or Kd, the meaning should be very clear to us. Now, let us take a look at isothermal titration calorimetry has been very successfully used to study protein small molecule interactions, protein carbohydrate interactions, protein lipid interactions, nucleic acid small molecule interactions, nucleic acid lipid interactions, protein nucleic acid interactions, nucleic acid interactions, antibody ligand interactions, receptor interactions, as well as non-biological interactions and others, including small molecule interactions. In short, if I say when anything is mixed with another thing, there is going to be some heat change. And if we are able to measure that heat change, we can connect that heat change with the mechanism of the process. And that is what our purpose here in these discussions will be, to understand the processes both qualitatively and quantitatively in terms of the thermodynamics involved in it. So, therefore, let us first now discuss the principle of ITC, principle of isothermal titration calorimetry. In an isothermal titration calorimeter, there is a sample cell and there is a reference cell. Constant power is supplied to both the cells to maintain the temperature difference between the cell 0, and everything is to be kept isothermal, because look at the name, the title, isothermal titration calorimetry. Unlike in differential scanning calorimetry, you remember in differential scanning calorimetry, the temperature was changed, the sample was heated at a constant scan rate. In isothermal titration calorimeter, temperature is not changed, temperature is kept constant. So, delta T is maintained 0, and then suppose if any reaction takes place in the sample cell, that reaction can be exothermic or endothermic. If the reaction is exothermic, temperature will tend to increase. If the reaction is endothermic, temperature will tend to decrease. Therefore, additional power is supplied to one of the cells to maintain delta T equal to 0, and that differential power is measured. That is what is commented over here. The differential power dP is measured power between the reference and sample cells to maintain a 0 temperature difference between the cells. This is the basic principle of isothermal titration calorimetry, and everything is protected by an adiabatic shield. Let me discuss in a little bit more details. What you have in an ITC is a sample cell, a reference cell, and in the sample cell an injection assembly can be inserted. As we can see here, there is an injection assembly over here. This is a syringe in which the ligand can be filled. You are filling the ligand in this syringe, and at the end of this syringe you see there is a small needle and there is small pedal over here at the end with a hole. This pedal will act as a stirrer because this entire thing can be rotated, and the solution, when the plunger is pushed, the solution can come out of here and get mixed with the contents of the cell. Same thing we can see in this figure in a little more clearer way, that within these adiabatic shields you have a sample cell, you have a reference cell, and in the sample cell you have this injection assembly inserted through which the ligand can be injected, and the end of this injection assembly is in the form of a pedal we can see here, and this entire assembly, that is that the syringe can be rotated so that as soon as a small injection is made and the solution is delivered outside, it is immediately mixed with the surroundings. So, this is how the things look inside the calorimetric cells. The reference will contain usually the buffer, and the sample cell will contain the sample of interest or the macromolecule of interest, and the syringe will have the ligand. Let me show another picture. The syringe can either be a fixed kind of syringe, that the whole assembly can come out of the cell where the syringe cannot be dismantled, or the syringe can be like this. And you can clearly see here this assembly, from this assembly the syringe can be taken out, that we can show in some other lecture, but you see here the needle which is extending out of the syringe has this sort of pedal at the end, and there will be a hole in the pedal through which the solution can be delivered. And that is what is commented in here. Ligand is taken in the syringe, macromolecule is taken in the sample cell, heat of interaction is measured, and the parameters which we will eventually get from a single ITC experiment are affinity. Affinity means the binding constant or equilibrium constant, binding mechanism, and number of binding sites. We will discuss the principle in a little bit more details. Now, let us concentrate on the sample cell and consider this as the sample cell in which the red, these moieties, take these as macromolecule in the ITC cell, and this is the injection assembly, and these dark circles, the black circles, let us consider these as ligand molecules in the syringe. And what we want to study is how these molecules, when injected inside, will interact with the protein, bind to the protein and lead to changes in the heat signals. Let us take a look at this. As we just discussed, microcalorie per second or microjoules per second, IUPAC suggests that we should talk in terms of joules, but many journals will accept even in calories. But whatever units we use, microcalorie per second is power. So, you see, initially when no injection was made, what you have is a horizontal line. Let me explain this in a bit more details. What we have, either microcalorie per second or microjoules per second, let me just represent microwatts. Or here I will have time. Let us say I also write this as microjoules per second. Initially, when no ligand has been added to the sample cell, no additional power is supplied, so the output is 0, means there is no differential power. No additional power needs to be given either to the sample cell or to the reference cell, so you get a straight line. When you make the first injection, that when you add a little bit of ligand into the protein solution, this reaction will start, P plus L, this reaction will start. And depending upon whether the reaction is exothermic or endothermic, additional power will be supplied to either the sample cell or the reference cell, or vice versa, means you can supply less power to the sample cell if the reaction is exothermic. The idea is to maintain the temperature difference equal to 0, and the entire thing should be kept isothermal. So, that differential power is monitored, and after the reaction is over, it goes back. Now, you see, y axis is microcalories per second or microjoules per second, x axis is time. Therefore, the area, this area within the line and the curve is nothing but it is heat. Q. Now, let us go back to the figure, and that is what I was saying, that after some of the ligand is added and it interacts with the protein, additional power or less power needs to be supplied to the sample cell, and it is observed in the form of a deviation, and after the reaction is over, it comes back to the original. Now, you see, each protein will have a binding site, or different proteins will have binding sites for different ligands. And if we take a large amount of protein in the cell and add only a little bit of ligand, the ligand will go and bind to the protein at the binding site. But since there are still so many other protein molecules available with the binding sites available, when the next injection is made or when some more ligand is added, it will again go and bind and saturate, eventually it will saturate all the binding sites. So, now let us take a look at when next injection is made, again there is a deviation, and you can see in the figure more and more these dark circles are interacting with the protein, but there are still proteins with free binding sites available, and you continue the titration, and eventually after all the binding sites are saturated, the amount of heat change towards the end is nearly 0. And you see the entire thing represents the binding profile. Area under each, area between the line and this whatever the peak, if we calculate all the areas and just plot the areas versus injection number, we will get binding profile, and area is equal to heat. And I will discuss this a bit later, that is how these areas can further be utilized to calculate the values of delta H. But let us first try to understand the principle very clearly. The principle is that when a small amount of ligand is added to a protein solution, either an exothermic reaction or endothermic reaction will take place. Accordingly, more power or less power will be supplied to the sample cell, and the differential power is noted at each injection, and that is what is seen in this figure, that at each injection the differential power is monitored, and when all the binding sites are saturated, you see overall the change is turning towards 0. So, this is a one complete experiment for the reaction of a ligand with a protein. Now, the question is, what experiments to be done before the analysis? Before we actually go for analysis, how many experiments need to be done? We are interested in understanding the interaction of a ligand with a protein to form a complex. P plus L is forming PL, and the associated change in enthalpy is delta H. That is what we want to experimentally determine. But let us examine carefully that the protein is prepared in buffer, ligand is prepared in buffer. So, therefore, along with this main reaction, that is ligand binding with the protein also interacts with the buffer, that is a small amount of dilution, and the ligand also interacts with the buffer when this is injected, that is a dilution. Therefore, heats of dilution must be determined and corrected. For a complete experiment, before the analysis of the data is done, the following should be done. The first experiment is you take ligand in the syringe, protein in the cell, and let us say the associated value that we measure is delta H. I will explain how to get this delta H. The second experiment should be ligand in the syringe and buffer in the protein, and what we will monitor, what we will measure is the enthalpy of dilution of the ligand, delta H dilution 1. The next experiment should be buffer taken in the syringe and protein in the cell. This is the dilution of the protein, and the corresponding change is delta H dilution 2. So, obviously, these enthalpies, these heat changes delta H dilution 1 and delta H dilution 2 should be subtracted from the main delta H to get the accurate value of the heat of reaction or accurate value of the enthalpy change associated with the process. Therefore, delta H, this delta H which is associated with protein plus ligand forming PL will be delta H main minus delta H dilution 1 minus delta H dilution 2. That is, we need to do these 1, 2, 3 experiments in order to get the value of delta H. And let me show in this figure that area under each peak is represented by this dot. See, it is kilocalorie per mole, or you can plot kilojoules per mole. Area under each peak is represented by this dot, and this is how the heat liberated varies. This is the main experiment, heat changes associated with the main experiment, and A is with the dilution experiments. And let us appreciate the heats associated with the dilutions are very, very small compared to the main heats, and that is how it should be, because the blank effect should be minimum. So, what do we get overall? If this is the binding profile, which is an integrated, if we plot heat evolved at each step against molar ratio is number of moles of ligand divided by number of mole of protein, we can get delta H is the total heat liberated, and we can get the binding constant. As I will explain in details later on, the slope is associated with the value of binding constant, and how this binding constant is extracted, that also I will explain in the next lecture. And we can get the stoichiometry of a reaction. You see, the stoichiometry here is shown in the same way as we discussed during the acid base titration. The stoichiometry, how many number of moles of ligand are enough to saturate the protein. And another example, the conversion of this area into the heat, integrated heat profile. Upper one is the raw data, following is the integrated heat profile, and the solid line represents the best fit to the experimental data. So, what we have discussed in this lecture is the principle of isothermal titration calorimetry. That is how the isothermal titration calorimeter measures the amount of heat associated with the interaction of a ligand with a protein. And also that it is the area under the each peak which represents the amount of heat liberated or absorbed. And when this amount of heat liberated or absorbed is plotted against time or against injection number or against molar ratio, lot of thermodynamic quantities can be determined. Which are those thermodynamic quantities? The enthalpy of reaction, the binding constant, stoichiometry, change in standard Gibbs free energy, and change in entropy, that too from a single experiment. All these things we will discuss in details in the next lecture. Thank you very much.